Today I'll be doing a bit of a different type of journal club. Be presenting a few papers. So all these papers are about differential detection differential expression using single cell data, single cell RNA-seq. So I'll start in chronological order, but after that I'll change a bit. So there's this paper from describing the muscat Bioconductor package. The first author is Helena Crowell, who at this point, maybe a few of you, has made quite a few Bioconductor packages. She's, for example, part of the spatial experiment paper and a few other ones. Mark Robinson, the last author of this paper, you also probably recognize him. He's one of the two authors, along with Ojlak, of the EDGE-R package. They have a really good paper from 2010 that talks about adjusting for, that is a calc norm factors function from EDGE-R. So has been around for quite a while, working with statistical methods for differential expression. At this point, I think Elena was a PhD student in Mark's lab. In this muscat paper, they go ahead and first of all, they develop a simulation framework that simulates a bunch of different scenarios here in figure 1b. They have a few scenarios where they say that the expression of the two groups, group A and group, is going to be equally expressed. So that means they have basically the same curve here with the mean, the same shape. They have another one here which is called EP. That is you can see the two curves are the same, but actually both groups have different, both curves have two modes instead of being unimodal over here. Now, the main we're interested in is the differential scenario where the two groups have the same curves, the same shapes, they're unimodal, but actually the mean is different between the two groups. But then they start looking at other more complicated scenarios. For example, here where if you compute them between blue and orange in this DP scenario, you have the same mean, but however, the shape is different between the two, where here blue has more expression on that first mode, whereas orange has more expression on the second mode. There's this DM scenario, differential modality, where the two curves have modes, number of modes, and then this DB scenario where here it has a single mode, it's going to be in the middle, whereas the one has two modes, they don't overlap at all with the orange mode. So it's more complicated scenarios. So test all these scenarios. At the time when muscat pre-printed and then published, a lot of methods for doing expression on single cell data were testing the expression differences at the cell level. So there was a question of whether pseudo, that means aggregating gene expression counts, was appropriate or not in these scenarios. And so in their supplemental table 1, they list a bunch of methods that they start to compare. And so some of these methods are going to be using pseudo-bulk data. However, there's also questions of what type of data should use pseudo-bulk. And then other methods are doing the analysis at cell. So for example, MAST, a method that was doing, it still is one method that can test differences in expression at the cell level. But for example, EDGE-R, bulk data, they have two versions. One of them, they're summing just the counts. Another one, they're summing scaled counts per million. Then they also apply limma-voom with counts, or what happens if you some counts with VST residuals. So they have a few scenarios here that they start to test. So in total, they have 16 scenarios. And this figure over here, in general, shows that the two methods where we care about are the black little star, which is the EDGE-R sum counts, and then the dark green star, which is limma-voom sum counts. Those two methods over here, in general, have good error rate control. So on the y-axis of this graph, we have the true positive rate. You want that number to be high. But then also they're looking at a false discovery rate on the x-axis, where they evaluate at 3 different thresholds. They say like, hey, let's run this method and give me any of the results that FDR less than 5%, less than 10%. So in these 3 lines here, your dots should be mostly matching the location of those 3 lines. And so in general, like EdgeR and limma are doing pretty well. I don't remember here the different columns, but here, oh yeah, here we get like the D scenario, where you can see that the 3 black stars and the 3 green ones are like, they're actually maybe a bit more conservative, where the actual false discovery rate is like lower than what they set on the thresholds, and the true positive rate is still pretty high. This is like when you adjust the p-values locally versus doing it globally. The local one is when you adjust the p-values by cell type for each cell at a time. The global one is when you do it across like all the cell types combined together. So the local one is the one that is making more sense over here on the top of figure 1a. Figure 1b has different panels for the different methods. I highlighted here the EdgeR1 and the limma-voom. And this one is a bit relevant to us. What it shows here is the number of cells that you have for every cell type. They don't call them cell types in this paper, they call them subpopulations. But yeah, basically by cluster, how many cells do you have? And they show that like if you reach 100, which is the yellow point, you're basically safe. After that, like, yes, there's an improvement, but the big difference is between 20 and 100. And so like, let's zoom in, for example, at EdgeR over here. These 3 blue points are like what happens when you have just 20 cells per cluster. Then the green ones are when you have 50. The yellow ones are when you have 100. And you can see there's a big increase from 20 to 50, a moderate increase from 50 to 100, and then almost no increase after that. Like, yes, increase a little bit, but not that much. And so boom over here, some counts, the 3 blue points actually much further below the EdgeR points in terms of the true positive rate on the y-axis. And then even the yellow points, some of them have a pretty low true positive rate over here. So in part, this paper is the origin of what we've been in OSCA, as well as in scRNA pseudobulk DGE, that EdgeR can perform better when you have like lower. If you have fewer cells, you're gonna have lower. That's one of the aspects of that. But if we also look at the supplemental material that they have, sorry. Yeah, here, this one is like different expression levels. The different panels here are how expressed the genes are. And if we focus on the first row, which is differential expression, you can kind of see a difference between the black stars, which are EdgeR sum counts, and the dark green ones, which are like limma-voom sum counts. So there's a big difference here, and that difference gets smaller as the expression of the genes. So that's the origin of that documentation that we've been seeing in several places. They also look at a few other scenarios here. What happens if you have like, exactly the same number of cells in all your groups? So what happens if you have like some samples, maybe start to have some differences? So they got quite a different aspects that you can change. They recognize that like results based on simulations are, well, they depend on how well that simulation framework is performing, right? And they recognize that simulating single cell data is very complicated. So you're gonna take some of these results with that caveat. Cool. Figure 3 here is a bit complicated to understand because it's like an upset plot with two bars on the top. You want of your things that are overlapping to be, actually, normally you want like the stuff that is overlapping to maybe be like of importance. Here, what they're showing is the blue bars are the things that were actually equally expressed. So they shouldn't have been detected as differentially expressed. So you can see the blue are like, in a case, they're basically like false positives. So you want your blue bars to be, smallest. Then the yellowish two bars, these are the differentially expressed results. The dark red, like the actual differentially expressed. There's a few other scenarios of testing. If we look at EDGE-R or limma-voom counts, they don't really have much that overlaps other methods. Yes, it has this point over here, this point over there, a few other methods. Most of them, and there's that point over there, but basically starting here, like, oops, that was not the right row. Mostly they overlap. They're just different, those methods in these three columns. Those three columns have no blue bars, and basically they're really detecting things that were like either differentially expressed or some of the other scenarios that they simulate. So that's good. Whereas like MAST over here, which is run at the single cell level, does find things like over here where there's a, not the biggest blue bar, but like if you look at the y-axis of that blue bar, that's around 1,000 or so genes. So that, some of these methods that are testing things at this level can have a lot of false positives. So in general, pseudo-bulking actually has pretty good error control. And they summarize this a bit on this figure 5, where they have the different 16 methods over here on the x-axis. They have a few different here on the y-axis, and they color them whether they're good results, intermediate, poor, or it is not. And you'll notice that for EDGE-R and limma-voom on the counts, basically they have in blue. Limma-voom here has 2 NAs, has one thing that is bad. I'm going to change colors. Has one bad, whereas EDGE-R has like one poor, right? Also, performance. You could argue like, well, these methods were not really developed to detect DP or DB genes, but they might still be of interest biologically speaking. So those two methods are pretty comparable against one another, and they're doing like way better than like some of these other methods on the right, where like you can see there's a more red. Cool. So before I read a little paragraph there, I wanted to highlight, I think, something else here, which is this supplemental figure 8. This, we've already looked at some of these plots where like are changing the number of cells per cluster. Supplementary figure 8 is the number of replicates per that you're looking at. And this is relevant to us because we have a lot of data where we don't have a very large number of replicates. And so for a low number of replicates, if we focus again on the difference between the black star and the dark green one, like 4 or 5, there's almost no difference here. But like for 2 replicates, is a bit of a, you can see that with this dark green stars being like much lower than the black ones. So if you have few replicates, basically if you don't have a lot of data, in terms of replicates or number of cells per cluster or things like that, EDGE-R seems to be performing better than limma-voom for these scenarios over here. So discussion, summarize what I just talked about in this paragraph over here, where they say the number of cells required to detect these changes depends on many factors, including the effect strength, right? That's how the expression it really is, the number of replicates, the number of cells per sample in each subpopulation, and like other properties about like the single RNA-seq assay. So that's a bit of the summary here. Now, the implementation of muscat is there's this R package over here that's on Bioconductor. And the main functions that you need to be using are the aggregate data function, which creates a pseudo-bulk. And then this function over here called, which is pb stands for pseudo-bulk, and this is for differential state, I think. This method over here, this function over here implements different methods, including edgeR, which is the default, as well as the limma-voom method. Now, there is a GitHub issue here from 2019 between Aaron Loon, the author of scRNA, as well as the author, or one of the authors of OSCA, orchestrating signal analysis with Bioconductor. And you can see this is the story or the origin of the Pseudobulk DGE function. You can dive into the code and they have differences between the two. But in general, scRNA Pseudobulk DGE is going to be really similar to muscat pbDD. So that's the muscat paper. It's the foundation of quite a few of the other papers I will be discussing. So this one is a preprint from March 2023. This one, the first author is Gabriel Hoffman. He's the author of Variance Partition, Dream. He's the main analyst for a lot of the sets developed at Mount Sinai, a lot of Panos Roussos from their group. Now, a lot of times, the data they've been working with is for the Common Mind Consortium data set. That's a scRNA-seq data set where the data was generated for different brain repositories, so that they had to deal with large batch effects. And then Gabriel Hoffman has been working on how to minimize those batch effects, how to like actually model that appropriately. So that's the background for a lot of the work that they've done. Dreamlet builds on top of Variance Partition, and the paper here uses the same type of simulation framework that muscat developed. Now here, Gabriel Hoffman and Panos Roussos, they're working with a data set that is in the thousands of donors. So that's their real data that they had to work with was like that big. And so they ended up simulating a super large data set. Now, the Dreamlet package implements of modeling batch true mixed effect models. So that's a little highlighted here in these figures too, where on panel C, we can see scenarios where they're modeling their diagnosis, which is a variable of interest, using an effect model for diagnosis, but also a fixed effect model for batch ID. And they're comparing that to a scenario where you model batch ID as a mixed effect model. So that's as a random effect, random intercept in this case, to be more specific. And so the Dreamlet over shown in red has like a low compute time. It's not as limma-voom, but it gets low. But like maybe the thing that we care about more here is panel E, where we have the false positive rate on the y-axis. You want that to be low, and we can see that the red bar here for Dreamlet Poisson is actually one of the lowest. Some other methods here, like for example limma-voom in particular, you can see that this blue line over here is pretty high, above the 5% y-axis line, and so they showed here that like limma-voom is going to have some trouble adjusting for the false positive if you're modeling batch ID as a fixed effect. It doesn't support mixed models. That's why you don't see a blue line on the right side here. Cool. So One of the reasons for this is if you're modeling batch ID as a fixed effect, let's say you have 8 groups, then you're going to have 7 by 7 covariates, and you're going to lose degrees of freedom, 7 degrees of freedom for residuals degrees of freedom, and argue that actually that's important for limma-voom. Limma-voom performs better when the residual degrees of freedom is higher. Now, there's just aggregating, like doing the pseudo-bulk. Dream implements a bunch of computational like new developments using like on-disk memory representation, things like that. And it's very efficient. That's why like in panel A, you can see that the red line is pretty low in general. So that's pretty nice. But like for our cases, we don't have super large data sets just yet. But we do have data sets where we have a bunch of batches. That's always the case. Sometimes we only have 2 batches, which at that point you could say like modeling batch ID is a fixed effect term, not the biggest, because you're only going to have like a 1 coefficient and lose 1 degree of freedom. But we do have scenarios that have like 8 batches or stuff like that. So if that's the case, then like Dreamlet might be useful for you. Now, the Dreamlet software implements like a really nice set of tools where you like say like, hey, I want to look at the voom graphs here. Like we see one per cluster or cell type. We want to look at variance partition per cell, just like either like the top genes or like this violin plots that we're used to. Later on, once you have the results, you can also make these volcano plots. Pretty nice if you want to summarize things as a heat map. So the Dreamlet package implements a lot of like nice tools for testing things. However, I'll note that I'll note something in Dreamlet, which is the adjusted p column over here is when you a study-wide false discovery. This is the global p-value that we were talking about in the muscat paper. It's not an adjusted p-value by cell type. So it's all of them together. And so you will probably have to separate your top table results by type, and then within that type, your raw p-values, and then adjust them in contrast to Dreamlet actually computes for you. So that's one thing to note in terms of the software. And like a lot of these figures in the Dreamlet preprint are showcasing the software. So you'll see like very similar graphs. Basically, they're analyzing different data sets. Here they're analyzing a COVID data set. Later on, they eventually analyze a much larger data set. Now, in variance partition plots, here, like blue, which is the disease state, is the variable of interest. And so we can see some genes like CLU and NFKB1A that have a lot of partition explained by this blue covariate, and they end up being some of the most differentially expressed genes here in that volcano plot. So that's something that you can in general expect to see, where like if you have some genes that have a high percent of variance explained by the covariate of interest, they should typically also be differentially expressed, unless the variance of the measurements is too large. So that data set, the variable of interest was like the one that explained the most variance. These data set might be a bit more along the things you would expect, where like the actual covariate of interest diagnosis is this purple one, is super low. However, for example, there's the gene PTPRG that has a larger purple bar than other genes, and this is one of the genes that shows up as differentially expressed. Now, the Dreamlet preprint has the graph over here, figure 5G, which shows that like on the x-axis you have the number of nuclei per donor. So this is like the number of cells per cell type, or like the mean of that across all of the cell types, against the average percent of variance explained. There's a correlation over here pointing with a small p-value. Later on, they show that actually there's basically a direct relationship between like how much data you have and how many differentially expressed genes you are able to detect, which we kind of had seen a bit on the muscat paper, or heard about on the muscat paper, but they talk about it over here and in the dreamlet preprint too. Cool. So let me just quickly check. Yeah, this supplemental figure 3 is probably the ones that are maybe more of interest to us. This panel B here, x-axis is the samples per batch. Now, the Dreamlet paper is talking about like super large data sets, but we don't have like the large data sets. Probably going to be on this range between 3 and 10 samples per batch. And here, the difference that you should be focusing on is the difference between red and blue. And so let's say we're like at 5 or so. This over here is the difference in the accuracy in this AUPR, the y-axis, where you want values, between modeling things with a mixed model, which is what Dreamlet, and modeling them with a fixed effect model, which is what like limma-voom can do, for example. And so when you have a number of samples per batch, maybe in some cases we have, let's say, samples per batch, there can be a pretty big improvement by using mixed model. So is one area for using Dreamlet. So again, if batches, then you probably should start looking at Dreamlet. And then they compare against like the muscat methods, and they show like in those scenarios with batches, Dreamlet is going to perform better. There's also some use of the stories PI, pi1, sorry, estimate, and a few other graphs, like one over here, where they show like the effect of like what happens with like some of the genes that have a lot of variance explained. They come from some of the cell types that have a lot of more data. So maybe I'm missing a few things, but like that's mostly what the Dreamlet preprint is about. So if you have batches, the point here is like you probably want to be using Dreamlet. Dreamlet is going to be using the voom framework, and maybe there's a bit of disconnect. Like, well, the muscat paper shows that like for low sizes you want to use HR instead of limma-voom. So that might put you in a tough situation if you have batches, you have a lot of and so like you might have to decide like, well, do I want to be able to adjust for batches more than I want to maybe deal with the advantages of HR at low sample sizes? So that's a bit tricky there. Now another preprint, also from 2023. This one is from December 2023. With some authors that I know, for example, David Ariso. He has been in the bioconductor world for quite a long time. And Jerome Gillis has also been quite active in the bioconductor world. So in this preprint, they're like, oh, well, there's been a lot of methods that test differential expression. So that's basically testing for differences in means. Right here it says testing differences in the average expression, so testing differences in the mean across cell types. And they say like, well, why do we want to test just for differential detection? So that is test for differences in the average fraction of the samples in which a gene is detected. And so their idea here is like, well, what if we just pseudo-bulk, not the actual counts, but take our count matrix at the cell level and ask whether we can make it into zeros and ones, where ones are going to be the cells where the gene is actually expressed, and zeros are going to be everything else, like where it's not expressed. And so you have a raw matrix of zeros and ones, then you can pseudo-bulk it. And at that point, it's quite natural to be using the binomial distribution. The binomial distribution is a distribution for like the distribution of successes of flipping a coin. So like either it's heads or tails. So that's what they end up testing over here. And figure over here is where they apply their method. The panel A showing the minus log 10 p-value for differential detection, their method that they just developed, against the minus log 10 p-value of differential expression. This y-axis over here is differential expression using muscat, pbDD, using edge R. And so in this scenario, there's a bunch of genes that have colors that are not black. The orange is like the genes that were just differentially expressed but not differentially detected. The green ones are like those are both differentially detected as well as differentially expressed. And there's a small subset of points that are in pink, which are the differentially detected only genes. So in this scenario, you could say like, well, their method is not actually leading us to many new genes that we consider of biological interest, just a handful of them. And let's see at some of these scenarios of the different genes. So here, like as a green example, we have B, which is a gene that is both differentially expressed. The y-axis here is the log 2 CPM. You can see there's a difference in these two groups. Then on the right side, we have the log of the proportion of basically that gene being expressed. And you can see that there's also a difference here. But then you have cases like the orange genes, which is panel C, where you see a differential expression difference, but not really a differential detection difference. Now, the ones in pink are like genes like the one in panel D, where like there's no difference in differential expression, but is a difference in differential detection. So this could be of biological interest to us. Yes, if you look at the mean of the expression is basically the same, but like what percent of the cells are actually expressing the gene, actually there's a big difference there, right? So you could argue that a case like panel B there, let's say that on the healthy group over here on the left side, when like out of all the cells that we have here, a large portion of them are expressing it, but they're just expressing it a little bit low, right? But on the moderate case, out of all the cells that we have, it's in like a smaller proportion, but actually at a level, right? So if you compute the mean of this and the mean of those means are the same, is what we see on the left side, but actually there is a difference in behavior because in the healthy cells, a large of them are actually expressing the gene, whereas in the moderate cells, a small of them are expressing that gene. So that could be of interest. And now this data set here has tons of signal. So you could argue like, well, you know, that might be a lot of work just for finding these ping points, but they run this on a few other data sets. Actually, I think it's on this. Yeah, so in the supplement, we have another data set where there's a lot less signal. And you can see the number of points is a lot less than the number of ping points. And so this could be happening in some of our data sets. Now this is also actually on muscat. Muscat implements both the pbDD function, but the pbDD. This is pseudo-bulk differential detection. And so if you use the aggregate data function, you have to change one parameter to say basically you're going to binarize the matrix before doing the pseudo-bulk, and then you can run the pbDD. Now, an important detail here, if you're running pbDD, is that internally, I mean, this is actually documented elsewhere and mentioned in the preprint, but they use the number of cells as information for like computing like a normalization factor. So they don't use factors in this. but they use the number of cells, which is this NC parameter over here. So the reason why I bring this up is that if you're going to run the pbDD function, you should not use as like the number of cells expressed. Even in that covariate, it shows up as explaining a lot of the variance in a variance partition analysis, because that information is used internally by the method in the implementation here in muscat. And so like the preprint will talk more about that. Now you'll notice that there's like points that have yellow centers, whereas other points have black. And that has to do with this stage R approach. A stage R is a paper from 2017 where the idea is if you are doing analysis, let's say at the gene level and also the transcript level, and then want to adjust your false discovery rate across both analyses, then you have to use like a stage approach. And so here they do run both the differential expression analysis as well as the differential detection analysis, and then use the information from those two analyses for the same, basically those two p-values, to then basically compute a stage-wise adjusted p. And so you can see over here that like there's some pink points near the border of the black points that no longer have yellow circles. That's because they end up not being significantly differential once you take into account that p-value distribution across the two analyses together. You also see some black points that then have yellow circles, but it's mostly the other scenario. You see like stuff that had color that now has no yellow circles. So they argue in that like that is an advantage, but if you were just testing out this for the first time, I would argue that the main thing that you'd be interested in is like seeing how many pink do you have, right? Is genes that are differentially detected that you hadn't seen before, then actually start checking some of them. Do they seem to have, are they like biologically relevant for this, for the biological question that you're answering? And so maybe they are, maybe they aren't, right? It depends on how you interpret the genes like in panel B, right? You could argue like, well, yes, maybe there's a different proportion of the cells expressing that gene, but maybe it's not. Maybe what's not important is whether like all of the cells are expressing that gene, but like actually how much of that, let's say, protein is being released, right? And so at that point you will look at like the mean expression that will translate like levels. Although we know that there's some issues in correlation of RNA to protein levels, but like you assume, and then that maybe that's what matters maybe for your system, right? Like how much of that protein is being released, right? Not whether it's like being produced by like cells versus not. So this method over here, pbDD, is implemented in the muscat package, and so that's another reason for learning about that muscat conductor package by Elena Crowell and co-authors. Now the last paper I'll mention today, this actually has been published, so it's not a preprint. It's from 2023. And you'll notice that like the author, sorry, I didn't mean to do that. Notice that the last author is Mark Robinson. So that's author of EdgeR that I've mentioned before. Also notice that Helena Crowell is a co-author of this paper. Lucas Weber, who we work with for quite a few years, is also part of this paper. But then the lead author is Simon Tiberi, who is now, I think, an assistant professor in Italy. And so like Simone is the one that has been really leading a lot of this work. And he developed a package called Distinct. And the goal, if you see a lot of these acronyms and them from the muscat paper, the goal is to be able to detect these different scenarios, right? Like the DB scenario, the DM, DP scenario. All of those scenarios were some of the ones that were described in the paper. There's a new scenario here which is the DV, differential variance. So same mean, but different variance. And so the muscat paper show that like EdgeR and limma-voom and like other methods have a lot of trouble detecting these scenarios of DP, DM, DB. And in this paper, in this distinct paper, Simone et al., they come up with a method for actually being able to identify these cases. And the statistical method is based on the cumulative density function, or sorry, cumulative distribution, where that's like when you start adding all of the points that you've seen so far in the blue line or the red line. And we can see here that the differential expression is like where like the curves are basically the same, but just there's an offset. This differential proportion, you see the curve is a little bit like flipped, if you like mirrored, if you want to say that. You can also do that a bit here in this DB scenario. And so like the distinct Bioconductor package develops like a full framework. I think it's a Bioconductor package for detecting those scenarios. And you can see here how they compare against EDGAR, limma, and a few other things, a few other methods. And so I haven't like fully read this paper yet, but I guess the logical continuation of the work, the Muscat paper, and it could be interesting to check it out. Now, yeah, so I guess in conclusion, if you have small samples, like a sample size or like a lot of cells per cluster, etc., the Muscat paper show that you will probably want to use EDGAR instead of limma-voom. The Muscat paper also show that pseudo-bulk methods are going to perform better than the methods based at cell level. And then the dreamlet paper or preprint that you have a batch effect, you'll probably want to model it using a random intercept. And that actually, if you have a, then you want to also use, because of as much all of the efficiencies that are implemented in Dreamlet. And then this is a preprint by Gillis et al. As the one that shows that actually you might just be interested also in biologically in some of the genes that are differentially detected but not differentially expressed. The team paper is now trying to solve some of the problems that the Muscat described as like well supported any of the methods at that in 2020. So that's the overview of all these papers. Of course, there's a bunch of details that I jumped over, but hopefully these will give you a bit an idea of some of the methods that exist that have been either preprinted or published, and all these papers are using a simulation framework for showcasing that their methods actually either respect or do well enough to adjust for the false positive rate and some other basically performance of the methods, and the accuracy of the methods. So with that, I'll end the recording.